and webinar in the series for the Lunch and Learn. So thank you everybody for joining. Today, I'm going to be providing some recommendations and advice on common laboratory workflows for studying cell metabolism and cancer. If you haven't already met me before, if this is the first time you are joining, my name is Lisa Sturcombe. I am a product manager here at Millennium Science and Jomar Life Research. I represent the Agilent Biotech plate washers and readers, as well as the ELISA portfolio. Just a quick outline on today's session. We're giving a bit of background into metabolism, some considerations when measuring the actual measurements themselves when it comes to metabolism, some tools you can use, and then I'll be going over some common assays and rounding us off with a summary. So first and foremost, what is cell metabolism? Cell metabolism, in its broadest definition, is a series of chemical reactions that occur to maintain life, and it consists of catabolic and anabolic reactions that are linked together in pathways, and those are driven by enzymes within those pathways along with nutrient availability. When it comes to metabolism, this image here is often what comes to mind when we think of metabolism, but the reality is a little bit more complex. And this second image here is, I guess, a more accurate depiction of what metabolism looks like. And there are several experiments that you can use when looking at metabolism. You can measure the rates of change for your metabolites or the metabolites directly. There are several classes that you can use, from amino acids, lipids, carbohydrates, so on and so forth. Another option you can use is looking at the reactants and products in cellular respiration, or the building blocks for DNA, RNA, so on and so forth. Looking at cancer itself as well, cancer is a series of and a complex heterogeneous collection of diseases. And yet, despite this diversity, there are a series of hallmark features that are consistent to cancer. And one of those is altered metabolism. So before we look at what metabolism is altered in cancer, I just want to briefly touch into normal cells. And so under normal conditions, in the presence of oxygen, these cells will metabolize glucose to pyruvate through glycolysis, and then to CO2 via oxidative phosphorylation. In the absence of oxygen, under anaerobic conditions, normal cells will redirect this pyruvate away from mitochondrial oxidation and generate lactate. Cancer, on the other hand, really relies on aerobic glycolysis. And so this uses the accumulation of intermediates in the production of macromolecules, which are required for proliferation as well as reactive oxygen species. But these metabolic changes also are vulnerabilities, and so they can be exploited clinically. Some considerations when it comes to your metabolic experiments in general. First and foremost, your cell culture media components. Do you require serum starvation, and what effect is that going to have on your metabolism? What concentration of glucose are you adding to your media? Location and timing. So at what point in time is your metabolite of interest being excreted in the media? Should you be using an endpoint assay, or perhaps looking at multiple time points as well? Are you measuring one metabolite? Would it be more prudent to look at multiple metabolites or organelles? Plating density. Definitely need to stress this. Plating density will affect, and I'm sure we're all aware of this, it will affect all manner of cell behavior and changes within the cell. So do you need your cells to be proliferating for your experiment? Cell-to-cell contact. Obviously, the more cells you have in the plate, the more glucose that's being consumed, the more waste that is being produced. Sensitivity or lack thereof. So is the readout that you have generated, does it accurately reflect your experimental question? And what's the abundance of the metabolite measured? Are dramatic changes actually required to be reflected for a change in metabolism? And what instrumentation should you consider to use to look at your assay? Because each of them will have different sensitivities and so on and so forth. So from plate reading to imaging to mass spec. And when it comes to live cell metabolism experiments, what time are the reagents integrated and metabolized in the cell, but also when is the background produced? Because this is going to point to the time in which you are going to read your experiment. Also need to consider that using cytotoxic reagents will have consequences to your cell viability and therefore your metabolic readings. Something you should also consider, there are several strategies to normalize your metabolic data. And when it comes to plate reading, first and foremost, I'm sure we've done many of these in the lab, is determining your total protein concentration through a BCA assay. You can also use DNA quantification. When it comes to the Agilent Biotech range, the Take3 is a really nice piece of technology. From this image here, you can see it allows you to do your micro volume analysis on a high throughput scale, and it is incredibly sensitive. We have like CD values around 3% off the top of my head when it comes to the Take3. So this is going to give you incredibly accurate data, which is it's very important when you are using this quantification for successive experiments. Got your metabolic glow assays as another example. Imaging, cell count for accuracy, viability, mitochondrial morphology are all assays in which you can utilize. I do want to circle back to cell count because it is the optimal method for normalization. And this image here on the screen is a prime example of that. So if we were to look at detection alone, we can see our areas of interest, but through adding the imaging as well, we have a more accurate picture of the reality of our experiments. We can see that we are having some cells being washed away, as well as false positives. So by normalizing your cell count, you're providing the most useful information to your metabolic data. And there are several tools that you can use to capture metabolic data. As the Agilent Biotech Product Manager, I'm going to be touching on a few of the tools that we represent to help you with your assays. First and foremost, the Citation 5. So the Citation 5 is a multi-mode plate reader, and it has an sophisticated imaging system as well. And it really does provide a global solution, gives you application flexibility and sensitivity. The Citation 5 has the patented hybrid optics that Biotech is known for, using both monochromator and filters. Again, flexibility and performance, along with your advanced microscopy. And the real advantage can be just illustrated here on the screen through hit picking. Using hit picking in the Citation 5, you can detect your areas of interest and then simply image those wells, which is going to save you a significant amount of time and data storage. Tool for capturing data number 2. Agilent, I'm sure we're well aware of the Seahorse XF. So what it will allow you to do is to simultaneously measure 2 major cellular energy producing pathways in your cells in real time. And this includes your mitochondrial respiration and glycolysis. And this allows you to better understand the connection between your cellular physiological processes and your genomic and proteomic data. The seahorse can be conjugated to the Citation 1 multi-mode plate reader and imager, and this serves as a QC check for your cell seeding, as well as you can use a cell count for normalization. Again, just reiterating the point I made earlier, cell count is the optimal method for normalizing your data. So you can see here from this image, the citation really does provide a very easy and validated cell count-based normalization to improve your data quality and interpretation. At the bottom of the screen with this data here, we can see the raw data, and this is your SCOV3 cells. And they were plated at 10, 20, and 30,000 cells per well. We have the raw OCR and ECAR, and we're looking at the change following the injection of oligomycin and FCCP. And by normalizing the data, we can see we have very nice, consistent, results. Lastly, I wanted to touch on the tool number 3, the Synergy Neo 2, and I could not talk about the Synergy Neo 2 when it comes to your fully detecting any metabolic assay. The Synergy Neo 2 is the flagship plate reader from the Agilent Biotech range. It was initially built in mind for the pharmaceutical market, but thanks to the flexibility of this instrument, you really can build it up or down to your needs, so it really will serve any laboratory and their applications. It has independent optimized optical paths, so it's going to be incredibly fast and sensitive. You're not going to be compromising or losing any signal with the Synergy Neo 2. It has our hybrid painted optics, again, that's Biotech is really known for, and it's using your 3rd generation quad monochromators in combination with your high performance filters. Really, this instrument does allow you to have uncompromised speed and performance for any of your assays. And there are many, many reasons for this. I'm just going to touch on a few right now. One of which is the dual PMTs. And this is really important when you're looking at your ratiometric assays, such as your Brett, FRET, so on and so forth. So with the dual PMTs, you're able to measure individually, but at the same time, your dual readout. So this is going to be incredibly faster than reading one at a time. And also there's no chance for signal crossover as well. So you're going to have incredibly accurate data quickly. You can build in laser-based detection into the Neo. So with, for example, this is the TRF laser that you can build into the Synergy Neo 2. And from the Z primes here, you see how accurate the data you can produce from the laser-based detection compared to a xenon light source. I'm just highlighting an example of some time-resolved fluorescence assays that you can utilize to examine metabolism. So in this visual abstract here, we're looking at oxygen consumption, and this is a metric that you can use to determine the effects of treatments on your cellular respiration. And how this works is by detecting how much O2 in the cellular microenvironment is being expended over time. An example of one such assay is the Agilent Mito-Express Extra Oxygen Consumption Assay. We have a illustration of the assay infographic just here. So using the Biotech instrumentation range as well as the Gen 5 software, you can define your data, your emission delay curves as well through there. Next, you detect your extracellular level consumption kinetically for both of your read steps. From that data there, Gen 5 then calculates the metric in the software, which is the lifetime value for each of these. And using this data, the OCR is computed as the slope of the linear response and New-Connect curve, all of which is done through Gen 5. And you can then report with your finalized data. So what we can see compared to your untreated cells, shown here in the purple bar, is that the addition of an uncoupling reagent drives maximum oxygen consumption, which we can see for the 2 data points in blue. Whereas when you disrupt the electron transport chain, you're going to have an inhibition of your oxygen consumption rate, which we can see in the bars in red, green, and green. We have no cells there. So moving on from oxygen consumption, there's also several mitochondrial measurements that you can utilize in your laboratory. ATP is a prime example since it is ubiquitously generated from oxidative phosphorylation, beta oxidation, and reactive oxygen species. And there are several commercially available kits along with imaging probes and mass specs. So several ways in which you can measure ATP. You can also use membrane potential, looking at fluorescent dyes for imaging. And a prime example of one of those dyes is from Biotium. They have a Mito-ID dye, and Biotium, if you're not aware, have a variety of fluorescent-based assays, high quality, longevity, great. And JOMA Life Research, which is joined with Millennium, does supply the Biotium, dyes themselves. In addition to that, you can have your typical cell viability or proliferation assays, such as MTT, which looks at the mitochondrial enzymes. So that's something to keep in mind as well. So looking at mitochondrial membrane potential, one such kit that you can look at is the Mito-ID membrane potential kit. And this is from Enzo Life Sciences. So this is engineered as an alternative to JC1, which is a a popular original Mito membrane potential kit. So the Mito-ID is much more water soluble than JC1 and allows for your live cell staining without washing or medium removal. And how it works is that it's capable of selectively entering into the mitochondria, and upon membrane potential increases, the dye will reversibly move from a green to a red state. So essentially what this means is that mitochondria with a low membrane potential will accumulate low concentrations of the dye and will exhibit a green fluorescence, whereas your more highly polarized mitochondria will exhibit an orange-red fluorescence. And here we see some data for comparing the JC1 and the Mito-ID. So because of the increased sensitivity, so I think the Mito-ID is about 10 times more sensitivity. It's also improved aqueous solubility and no wash requirements. We have incredibly high Z factors compared to your JC1. And because it is the only HTS assay that monitors membrane potential, that doesn't require washing, it can be used in real time. Following on from your membrane potential, looking at metabolic stress and autophagy measurements is something that you can look at as well. So autophagy itself is a degradation and recycling pathway, and it's essentially under undesirable conditions allows the cells themselves to adapt, and so it allows them to promote cell survival, but ultimately it can lead to death. There are a number of strategies and kits available from fluorescent dyes that can be used in imaging, flow, or plate reading when it comes to autophagy. Two ones that I just want to briefly mention here from Biotium. You can have the lyso-view and the lipid spot stains, but I will go into a few other ones in a little bit more detail. Abcam, I'm not sure if you're aware, JOMA Life Research has recently taken on Abcam as a supplier, so we do support everything other than their primary antibodies. So the autophagy kit can be used to measure your autophagic vacuoles and monitors the flux in live cells. And it can be used for flow, microscopy, or detection. We have an example of your data here. We have some cells that are being treated with rapamycin, and we can see the increase in the actual kit and the kit and the data itself at with increasing rapamycin concentration. In addition, we have the Mito-ID 405, which rapidly stains mitochondria in live cells, again, not needing washing for this. And it changes localization upon mitochondrial depolarization. Another one to focus on briefly, and the last one for today, is the Cyto-ID autophagy detection kit from Enzo Life Sciences. It rapidly quantifies autophagy in native heterogeneous cell populations. It is a cationic amphiphilic tracer, or CAT dye, and it rapidly partitions into cells to induce phospholipidosis. An advantage is that it doesn't require transfection, so it's going to significantly save you time. It's also specific. It differentiates between autophagic flux and autophagosome accumulation with negligible backgrounds, with us a whole host of features. So looking at some data here, we see we have Hep G2 cells. They were treated with tamoxifen for 16 hours. Tamoxifen increases your intracellular ceramide levels and abolishes the inhibitory effect of PI3 kinase. From the results here, we can see an increase in Cyto-ID signal from tamoxifen concentrations from 1 micromolar. We then have Vipramil. It's the same cells, the Hep G2 cells, and Vipramil passively diffuses into the lysosomes and becomes trapped upon cremation and causing an increase in liposomal pH. And we can see an increase in Cyto-ID from about 10 micromole onwards. So just coming to a summary now. So what we can see is that metabolic changes represent a broad but representative tool to study cancer as well as cell biology in general. And this, there's a diverse range of metabolic targets that we can use to examine or exploit clinically. And lastly, but not least, multi-mode readers and the biotech range have a diverse, comprehensive, and flexible platform to evaluate cellular metabolism. And it's a rather inexpensive technology that really does offer a myriad of flexibility and diversity to address your research questions. Lastly, I just wanted to thank everybody for their time, for joining today, as well as for this series in its entirety. I understand everybody is very busy, so I do appreciate you taking the time to sit down and have a bit of a chat with me about all things to metabolism. Now I want to open the time for questions, and if you don't have a question now, my email is on the screen, so feel free to contact me at any point in time. I will leave the floor now for questions if anyone has any. Thank you. no worries whatsoever. If that's the case, we might finish things off early. I'll give you some time back into the rest of your day. Feel free to look at our websites or contact me if I can be of any assistance to you. Enjoy the rest of your day, guys, and I'm sure we'll be in contact in the future. Bye.